Hi, my name is Mary Masako Axhelm, and I'm about to discuss some things in my first lab video for microbiology. One of the things is my calendar and keeping on top of our labs, seeing as we have 2 for the next 3 weeks. So I've written down some things on a calendar in regards to the pouring plates, the incubation period, the starting of the cultures, and the observation. So I'll be going over that briefly and also talking about my lab kit, which is pretty extensive. And I wanted to talk about that. I had a little issue with something on the broth, one of the broths that came in. And also wanted to discuss my incubation place where I found in my home. I think it's sufficient. The temperature seems to be right. So, and then go over a couple of tips and tricks and cleaning up and stuff like that as far as the labs go. So, nice to meet you, and here we go. Okay, so the first thing that I did was I took a calendar and I noted, we started on this day, but I noted that our lab 1 would be due, and I numbered the labs because they actually were not numbered in online. So numbering them really helped me actually, and in turning my assignment you'll see that I numbered it as well. So for the next thing that we're going to be doing, I'm going ahead and taking lab 2 and 3 and starting the pour plates. I hope I said that right. Then the incubation period will be starting around here. For lab number 2, incubation is 3 to 6 days. Lab 3, 1 to 3 days. So I'm assuming or kind of gauging that observing the results will be around this day. And then on to observing the results for lab number 2 would be this day, and then starting the following week, which would be labs 2 and 3 being due, and then starting the culture for lab 5, which would be about 1 to 3 days. There's no incubation period on that, and then observing the results. So basically starting a pattern of overlapping, turning in something, and then also starting another, and then keeping track of the incubation period and observing the results. So that's for July, as you can see. I also did it for August, same type of situation, observing results for lab 8, then lab 8 being due, starting the culture for 9, incubation, observation, and then the pour plate. We didn't have very many of those. I think this is the second one. Then lab 9 being due, starting the culture, incubation, and then observing results. Lab 10 being due, and then this being the last week of our class. Alright, so on to, have everything out here, on to the lab kit. I took everything out and I checked everything off. So everything seemed good to go. As you can see, I made check marks. Made a little note that didn't say wipes before. There was one thing. This is it. It's ASSY MBK culture bag number 3. These are 6 broth, and it said phenol red 9mm. It was leaking. I'm going to go ahead and show you. I haven't put it in the refrigerator yet, but put it in a separate bag. It actually looks like one of the broths is low. I don't know if you can see that, but it was leaking inside the bag. I put it in a Ziploc bag because I didn't want it to get all over the place. But, so this is the situation, and I'm not sure what do you think I need to do. So you can see the levels are really low on that one, and the other ones seem to be okay. I'm gonna go ahead and refrigerate it, but let me know. I'm gonna also email you, Professor, to find out what to do. But everything's here. I went over the refrigerate notice, so I'm gonna put that stuff in the fridge. Emergency contact, there's also safety information on the back. And I have taken physiology and anatomy, so I do know about cleaning up with the 10% bleach solution. And so I will definitely use my goggles as a safety precaution. There's quite a few things that I can do, such as washing hands. It's really important. Using gloves when I'm working with specimens or cultures or whatever is going to be, whatever I'm going to be working with. It's really important to not have pets or children around. And I do have a child who's 12 years old. I have no pets here except for a hamster, but I'm going to make sure that I am working in an environment where I'm not going to be disturbing or causing children to be around. So, yeah, so wearing the protective eye gear, keeping the cultures and laboratory materials out of reach of children and pets, clearly labeling what needs to be labeled, and also cleaning up after myself. I think it's really important to wash hands with soap and warm water before and after. I put on the gloves. If anything spills, it will be picked up immediately and using a 10% bleach solution. And I will be disposing of the plasticware, but first soaking it in a bleach solution probably for about 2 hours, wrapping with paper towels, sealing in a plastic bag just as I did with physiology, and then putting it in the garbage. Yeah, so mostly everything will be put through a bleach process before it's thrown out. I am concerned about working with certain cultures. I'm allergic to penicillin, but definitely going to take precautions in everything that I come across, and I'll probably ask you questions as we go. And I also wanted to mention about the incubation area. I'm going to go ahead and show you where I've decided to do that. Let's just go this way. Sorry, my trash can. So that's where I'm going to do it. I have a really small apartment, so I don't really have anywhere else to do the incubation process. My biggest concern, I think, is the drafts, because I have central AC and it's been really hot lately, and we've got ants, a lot of ants, and I've been trying to get rid of them with all different types of things, like the taro treatment and borax. I'm going to try that next, but that's become an issue, and I don't really want to draw more ants into my house, and so I'm trying to keep it cool as well. So hopefully that will be satisfactory, and I hope you liked the video. I didn't want to go too long, so I'm going to cut it out here. And I think I covered all the bases. Thank you so much. Bye.